you're going to generate new electron carriers. You're going to load them up with electrons. And then you're going to attach this coenzyme A to it. And so after this transition period, you end up with acetyl-CoA. And it's that acetyl-CoA that can feed directly into the citric acid cycle. So we've completed glycolysis and the Krebs cycle. So we've completed the breakdown of glucose. We've stripped all these electrons away from them and we've given them to our NADs and our FADs. That's all that's happening in glycolysis and the Krebs cycle. So once we've loaded up all of our electron carriers, now we're going to give them to oxygen. So we do that in the electron transport chain. And so all of these NADs and all of these FADs will come and they'll be oxidized because they're giving those electrons away to the proteins in the electron transport chain. And so as those electrons move down, they're going to interact with oxygen in order to form water. And then finally, you have chemiosmosis, which is again due to this active pumping of these hydrogen ions outside of the matrix. And the diffusion will bring those hydrogen ions back in, and it's that gradient that's used to power ATP synthase so that you can make lots and lots and lots of ATP. And the way that the ATP is made in oxidative phosphorylation is a process called oxidative phosphorylation. All right, so two ways to make ATP, either substrate level phosphorylation or oxidative phosphorylation. Oxidative phosphorylation is going to be way more efficient than this substrate level phosphorylation. Questions? All right, let's see if we have any more slides. I don't know if we do or not. Oh, we do. Okay. All right, so that's cellular respiration. So when we're talking about cellular respiration, we're talking about aerobic respiration, right? So respiration with oxygen. And again, anaerobic respiration, that oxygen is always going to be that final electron acceptor at the end of the electron transport chain. So now we're going to move on and talk about what happens if you don't have oxygen. Well, if you don't have oxygen, you're not going to have that oxygen at the bottom of the electron transport chain. And so this is why it's called anaerobic, because it's without oxygen. And so in these conditions where you don't have oxygen, you're going to have some other molecule that's going to serve as the final electron acceptor. Sulfate is another molecule that can serve, and if my memory serves me correctly, sulfate is SO4, I think, something along that line. So it's a sulfur and a couple oxygens in there. But anyways, in anaerobic, this might be sitting, so it's still got oxygen in it, it's just not oxygen by itself, if that makes sense. It's not like O2 oxygen, it's just sulfate. Anyways, the final electron acceptor in anaerobic respiration is going to be something other than oxygen. And then there's an alternative method that are used by some organisms. It's called fermentation. And so this fermentation process is going to use this phosphorylation in order to generate energy. So in fermentation, you're not having, there's no Krebs cycle, there's no electron transport chain, there's no chemiosmosis. In fermentation, you're starting with pyruvate, and you're ending up with either alcohol or lactic acid. And so one thing that I don't think I've mentioned yet is that glycolysis is universal to every organism. All right? So every organism, doesn't matter if it's anaerobic respiration, aerobic respiration, or fermentation, they're all going to use glucose. And they're all going to use glycolysis, which means you're always going to have the breakdown of glucose into pyruvate. So pyruvate can feed into aerobic respiration, anaerobic respiration, and fermentation. And so we're going to go and compare these. And so there's two different types of fermentation. And again, fermentation is, the process of breaking down sugars and releasing carbon dioxide. That's essentially what we're doing in cellular respiration. Except that this fermentation, this is going to be primarily seen in bacteria and yeast. All right, this fermentation process, this is how we make, our cheeses, our yogurts, our wines, our beers. This fermentation process is used by us every day, or not us directly, but we come in contact with the products of fermentation every day. And so there's two different kinds. You've got the alcohol fermentation. This is the type of fermentation that's used to make, beers and alcoholic beverages. And then you also have lactic acid fermentation. And so both of these are going to start with pyruvate, but in alcohol fermentation it's going to be converted into ethanol, and in lactic acid fermentation it's going to be converted into lactic acid. And lactate is the exact same thing as lactic acid, so don't get confused by that. It's just, we love to have 3 trillion names for everything, right? So lactate and lactic acid are the exact same thing. And so this lactic acid fermentation, this is used, to make the cheeses and the yogurts and a lot of other dairy products. And actually, we are actually capable of using lactic acid fermentation. So if you've ever, gone and worked out, you're running like 6 miles or something, you start to feel that burn. That burn is a buildup of lactic acid. All right? It's acidic, so it's going to burn a little bit, right? And so what's going to happen if you're, not stressing your body out, but if you're actively using a lot of energy, there's obviously still oxygen present, we're still breathing, but there's not enough to keep up with the energy demand of your body. And so what's going to happen is your body, or particularly those cells, those muscle cells that are being affected, they're going to switch from aerobic respiration into this lactic acid fermentation. And so it's doing this because this process doesn't need oxygen, right? So there's not enough oxygen to go around to generate enough energy through cellular respiration. So your body will switch over to a backup mechanism for producing energy, which is this lactic acid. And so as a result of this lactic acid fermentation, you're going to get the formation of lactate. And it's going to build up in your muscle cells, and that's what gives you that burn that you feel when you've been, exerting yourself physically. All right, so anyways, let's go back and let's look at both types of fermentation. So the top one is going to be alcohol fermentation. And again, both of these processes beforehand, glycolysis is occurring, so you're always going to end up with those 2 pyruvates. Now in fermentation, that pyruvate molecule will be rearranged into various different molecules in order to produce ethanol. And so the products of alcohol fermentation are going to be 2 carbon dioxides and 2 NAD positives. And so one of the other points of this fermentation process is to regenerate these empty versions of the electron carriers. And so because we are basically breaking down pyruvate again, we're removing carbon dioxide, and in the process we're giving those electrons away so that we can generate more of this NAD positive. And then at the end we end up with 2 ethanols, which ethanol is just a 2-carbon alcohol. Now in lactic acid, again, you're still going to have glycolysis, so you're still starting with pyruvate, but pyruvate is, again, going to be converted into lactic acid or lactate. And then same thing, we're also generating, because we're taking these electrons that we generated, or these electron carriers that have these electrons from glycolysis, and some of them are going to be used to make lactate, and some of them are going to be used to make ethanol. And so in the process, you're oxidizing this NADH in order to generate the NAD positive. So the products of lactic acid fermentation are going to be 2 lactic acids and 2 NAD positives. The products of alcohol fermentation are going to be 2 carbon dioxide, 2 NAD positives, and 2 ethanols. All right, so you're still generating ATP through this process. It's just going to be less efficient. So each one of these processes is going to make 2 molecules of ATP. And so you can see that fermentation is definitely not as efficient as cellular respiration or aerobic respiration, because you're only making 2 ATPs here, whereas in oxidative phosphorylation you're making 32 to 34. So this fermentation, as far as humans are concerned, this is just kind of a backup mechanism for generating energy. So when we're physically exerting ourselves, a lot of times we'll turn to this lactic acid fermentation. But other organisms like bacteria and yeast, they are only one cell. They're unicellular organisms. So they don't need this big complex cellular respiration. You know, they're only caring about one cell. And so they can get away with using fermentation to generate their energy. Questions? Yeah, who's your?
So for the exam, we're supposed to know basically, like, for example, alcohol fermentation, it starts with pyruvate and then it generates 2 ethanols, 2 NAD positives, and 2 carbon dioxide.
Yeah, and 2 ATP. They're originating from this glycolysis, because fermentation is always going to be tied to glycolysis, just like any other type of fermentation is. So, I mean, technically it's the glycolysis that's producing the 2 ATP, but they're linked together. So you throw in 2 ATP in there, because you're still, the whole point of this is still making energy, just doing it in a different type of fashion. Other questions?
Sorry, ATP is going to be included in the product.
Yes, ATP is going to be included in the products, yeah. Because the whole point of fermentation is just an alternative mechanism to generate energy. And so it wouldn't make sense to say we're using this to make energy if energy wasn't in the products. So yes, throw ATP in the products as well. Other questions? Okay. So this is kind of comparing all the different types of respiration. And again, all of them are going to use pyruvate. All of them are going to use glycolysis, which means you're always going to have pyruvate. And so if oxygen is present, that pyruvate will go through this transition period, be converted into acetyl-CoA, go into the Krebs cycle, go through, oxidative phosphorylation, go through the whole process. All right? If it's anaerobic respiration, that final electron acceptor is going to be something other than oxygen. If it's aerobic respiration, that final electron acceptor at the end of the electron transport chain has got to be oxygen. So this process is the same for aerobic and anaerobic respiration. And the only difference is what that final electron acceptor is. If it's oxygen, you know it's aerobic. If it's not oxygen, it's got to be anaerobic. All right, so let's say you don't have any oxygen and you don't have the capability of using anaerobic respiration. So an alternative mechanism is this fermentation. And so when you don't have oxygen, you can feed into fermentation, either the lactic acid fermentation or the ethanol fermentation. And again, the products of these the main products are going to be ethanol and lactate. You're also regenerating some other stuff, but I mean, the main products are going to be ethanol and lactate. But again, glycolysis is going to occur no matter what. No matter what type of cell, no matter what type of organism, no matter what type of respiration, glycolysis is always there. Okay, so this is kind of what we've already been talking about. So fermentation and aerobic respiration are both going to use glycolysis. And the difference between the two is that that final electron acceptor is going to be different. Again, in aerobic, it's going to be oxygen, and then in anaerobic, it's going to be something other than oxygen. All right, any more questions, thoughts, or concerns? Okay, I will see y'all on Tuesday.